Hi friends, today we'll be talking about the different what you call as solutions which we prepare in the laboratory. Many of the times like you go in the laboratory and you have to prepare what you call as some dilute solution from the stock solutions which are already available in the laboratory. Now like for example, if you go in a lab and you have some 80% of a particular solution and you have to prepare 50% of that, you are having 90% of a stock solution, you have to prepare 20% of that. And many of the times then we get confused, like what exactly I'll do now. First thing which I would tell you that please don't bother much, it is very easy. Many of the times like we feel that we have to do a lot of mathematics and we go for solving it in a very complicated way. I've seen many books and I've seen many places where they make it so serious issue that students, they start thinking that it is something out of the world. So let us first come to the conclusion that making a particular dilution from a particular stock is something which can be done very easily. So let me be with that first, right? So with this, like with a very small example, let me explain you that if you have a stock solution of a particular concentration and if you want to prepare a dilution of that particular stock solution, then how exactly you will go with it. Now it becomes like it varies, like you're going from molar solutions to normal solutions, or you are preparing percent solutions, right? Today I'll be talking about the percent solution. In upcoming videos maybe we'll talk about some other ways of expression of these concentrations, but remember the crux, it remains the same. Now say for example, if I come to my laboratory and I found that I have around 80% of ethanol solution which is there as a stock in my lab. Now I came in the laboratory and there was this 80% of ethanol which was present, and my lab instructor, like he or she came in the laboratory and he told like go ahead and prepare around 40 ml of ethanol which is having a concentration of 60%, right? So I have a stock solution of 80% of ethanol, right? I have to prepare 40 ml of ethanol with 60% of the concentration. This is what I have to do. As I told, first thing which will happen to me is what the hell I will do with this, right? How will I go from 80 to 60? Whether I take 2 ml, whether I take 5 ml, I'll go and search everywhere and I'll go on asking people and I get confused and I say that this is very difficult. So as I told, don't do that. Simple formula. Remember, it goes like this. It is C1V1 is equal to your C2V2. Right, simple. So it is C1V1 and it is your C2V2. Remember this. So C stands for the concentration and V stands for the volume. Right. So you have one particular solution, you know the volume as well as the concentration, both. अभी आपको दोनों पता है, मतलब you know the volume and the concentration of both. Which one is that? You know this, you want to prepare 40 ml, you know this, that its concentration is 60%, right? So I'll put in here the concentration is 60, right? The volume which I want to prepare is 40, right? So the second one, I have 80% of the ethanol. I don't know how much of that ethanol I am going to take to prepare the final concentration of 60%. So there I'll take a concentration of 80 and let this remain as V2. So simple calculation I can do now. So this will be 60 into 40 upon 80. Right, can you please help me with this? Right, this will become how much? 6 fours would be 24. This would be 2400 upon 80. Right, so this would be like 30. So the volume which I got is around 30 ml. Now what exactly I'll do is from this particular stock solution, I will take 30 ml. Right, I'll add 10 ml of distilled water, right? And this will give me a concentration of 60% with a volume of 40 ml. It is as simple as this, right? So remember the formula, C1V1 is equal to C2V2, right? You know the concentration, you know the volume. You know the concentration, you calculate the volume. Now you know the volume, you also know the final volume which you want. Subtract it and now add that much amount of your distilled water and get your solution. So stay tuned with me for more in biochemistry, molecular biology, practicals and theoretical aspects in microbiology and molecular biology. Thank you so much.